You might be familiar with biologists adding genes to plants for larger fruits, longer shelf life, and disease resistance. But researchers are considering more complex applications with plants, allowing them to switch traits on and off in living cells or use plants as life-saving environmental sensors. This development is often called synthetic genetic circuits. At first glance, there's nothing in common with plant genes and digital circuits. The phenotype of a plant comes from multiple interacting genes and the environment. Instead of a digital on-off switch, conditions such as drought gradually shift plant cells' natural behavior depending on time and intensity. This is one paper's attempt to draw the cellular system as a circuit. When biologists are adding new genes, they can design more direct switches, which combine into circuits with controllable behavior. I first saw an example of this on the Science IRL channel, where the Center for Research on Programmable Plant Systems inserts a gene which activates under red light.
I built a light-inducible gene expression system where we can turn a gene on with red light and then immediately turn it off with far red light.
We're still at such an early stage, a lot of the research is just around testing what genes and promoters work as reliable logic gates. In crop science, there's the possibility these circuits could convert each plant into sensors, detecting contamination or undesirable chemicals. There's been interest in plants which could be seeded over minefields and indicate where these landmines were buried. Unfortunately, these have yet to be adopted in the field. In the 1990s, a team at Oak Ridge National Laboratory investigated bacteria which would respond to RDX and TNT in the soil. Their experiments had some false positives, but they were generally accepted. In 2004, Nature reported that a company had produced plants which changed color in the presence of landmines near the surface. Field experiments in the Balkans showed disappointing results in 2008 and 2009, and the company no longer exists. Fluorescent landmine detection bacteria continue to appear with new approaches and studies. Here's an example from 2017. There's an increased interest in genetic circuits that would allow plants to respond to the variable conditions of climate change. The same seeds could be used on a very large area and respond to the actual conditions in different fields or different parts of a field. While this is developing, a lot of research is going into finding out the features of genetic circuits. For example, in a plant or bacteria which goes through multiple generations of genetic recombination, how do you keep the circuit inherited intact? How could we use signaling in between cells to make more complex circuits and sensors? Can we program in memory to pick up on longer-term trends? Should we be making these circuits perfectly work switching on and off? Or at scale, should there be a strategic level of randomness to the behavior? This is going to be probably the shortest video that I've done in a while, but it's an intriguing topic which is just developing in plant research, and I hope this gives a good overview without just sitting and reading a scientific paper to you. There's links to everything in the description. Feel free to leave a comment, like, or subscribe. Thanks for watching.